Hello friends, this video is on color vision and color blindness. What is the importance of this topic? At the first prof level, there may be a short note on either color vision or color blindness. And later on, as clinical application, there are industries where you need to identify the colors properly, or motor vehicle operating, that's when you need to have a proper color vision, or there are certain drugs whose side effect is that they affect the color vision. So there is a lot of clinical application which we will also talk about it later on in the video. Let's start with first the physiology of color vision. Well, if it is a question in the exam for 5 marks or maybe even 10 marks, then you can divide it as color vision 5 marks, color blindness 5 marks. That's how you can attempt the question. And it should be under these headings: primary colors and the cones which are responsible for colors, then theories of color vision, then you talk about the color blindness, and then tests to detect the color blindness or defects in the color vision. So let's start with the introduction. Color is a subjective phenomenon. Yes, you might have seen certain photographs where you are asked to identify which color is that photograph, and you will see that two different people, they quote two different colors. So yes, it is said as optical illusion, of course, but then yes, it is possible because color is a subjective phenomenon. What are the various properties of color? There are three main properties of the color: hue, saturation, and brightness. Out of these, the actual color, means the wavelength of light, is the hue. Hue is the wavelength of light or the actual color. And then brightness tells us about the intensity of that color, etc. Now basically there are three primary colors. Well, from the artistic point of view, the primary colors are said to be red, yellow and blue. But from the visual spectrum point of view, from the spectrum of light point of view, there are three primary colors: red, green and blue. So remember, primary colors, when you are painting, that time you say as red, yellow and blue, that's fine. But actually speaking, when you talk about the physiology, you're talking about the spectrum of the light and wavelengths of various colors. That time primary colors for physiology, they are red, green and blue. So that's the point number one. This is the introduction. By the way, I've written the color spelling as COLOR. Please don't mind. I mean, it is COLOUR, of course, American and British. So let's go ahead. Now, you know that the photoreceptors are present in the retina. Rods are concerned with dim light vision, and cones are concerned with bright light vision and the color vision. So cones are the photoreceptors that colors basically. And then you can draw the diagram of a cone. Cone has got four parts: the outer segment, which has color sensitive pigment, and then inner segment, nucleus and synaptic body. So once the processing of the light has been done by the outer segment, then there will be neurotransmitter released at the synaptic body, and that is how the stimulation of cone would convey to the optic nerve fibers regarding the color sensation. Now cones basically are of three types: red cones, green cones and blue cones. And they are called so because of the pigment, photosensitive pigment present in their outer segments. So red cones will have erythrolabe, green cones will have chlorolabe, and blue cones will have cyanolabe. Erythros is red, chloro is green, and cyano means blue. So three cones that perceive these three primary colors. Now that brings us to an important section: theories of color vision. There have been various theories put forward as to how we perceive the color vision. Uh, the older one is the Granit's modulator and dominator theory. If you are writing this in the paper, you write this, you get additional marks. According to this theory, there are two types of ganglion cells in the retina: dominator and modulator. Now, this dominator ganglion cells, they are stimulated by whole of the visual spectrum. Entire visual spectrum stimulates these cells, dominator cells. So they will convey the intensity of the color and intensity of the light reaching the retina. Whereas the modulator, the other type of ganglion cell, it is stimulated by a very narrow wavelength band, and that is responsible for different color sensations. So these cells, modulator cells, they are responsible for different color sensations because they are stimulated by a very narrow wavelength, which is for a particular color. And dominator cells are stimulated by the entire visual spectrum, so they will convey about the intensity of the light and intensity of the color. That's one theory. Another theory is color opponent cells. These were all the hypotheses that have been put forward. Most acceptable theory nowadays is the third one, which will come to that. But color opponent cells, these are again ganglion cells in the retina, and there are various types of ganglion cells. For example, RG cells, red, green. So they are excited by red and inhibited by the green. So they are called as red on, green off, or by the reverse, green on, red off. Similarly, there are other types of ganglion cells like red on and blue off, so RB, and green on, blue off, so GB. And these are the various types of ganglion cells, and they are stimulated by one color. And opponent theory, color opponent theory means the other color, which is said to be the opposite color or opponent color in a sense, it inhibits those cells, particular cells. This was another theory. And of course, then this was in the retina, even in the lateral geniculate nucleus of thalamus. You know, from the retina, visual pathway starts and it relays in the thalamus, lateral geniculate body. Even there are color opponent cells. They process the signal from the corresponding ganglion cells. So this is called as color opponent theory. We have discussed two theories so far: Granit's modulator and dominator theory and color opponent theory. But the real theory that is in use and that is accepted today is Young and Helmholtz's trichromatic theory for color vision. So this will be the most important part of your answer. Young and Helmholtz theory, trichromatic theory for color vision. So trichromatic, three colors. Basically, as per this theory, there are three cones in the retina. We already saw about those three cones: red, green and blue. And there are three primary colors, therefore, which are detected by those three cones. Now, according to this theory, these colors have specific wavelength of the light. They are at a particular spectrum of the wavelength. So, red 570 nanometers, green 535 nanometers, blue 445 nanometers. Or you can remember it in the reverse: 445, 535 and 570. And then you can draw this graph. You can see here, this graph tells you about the absorption of a particular wavelength of light, or a particular light with a particular wavelength. For example, you can see here on this side, violet and blue color light. If it strikes the retina, then it will be maximally absorbed by the blue cones. Then 535, in that range, green cones. So if the light wavelength is of 535 nanometers, then green cones will absorb it maximally, and red cones will absorb the light maximally if the wavelength is 570 nanometers. And that is how, from their absorption, from the absorption of light by various cones, we detect the primary colors. So that's the Young and Helmholtz theory of trichromatic theory of color vision. This is just a diagrammatic representation of how we perceive the primary and secondary colors. So talking about the primary colors, ratio of stimulation of three types of cones based on the wavelength of light. Let's say light wavelength is of let's say 570 nanometers. Now this light wavelength will maximally stimulate red cone. This is a retina and you can see three different cones. So if the wavelength of light is in this range 570 nanometers, then it will maximally stimulate red cones 100%, and green and blue cones will not be stimulated at all, 0%. And this set of values, this set of stimulus strength or stimulus values for the cones 100, 0, 0 will be perceived as the monochromatic red light or red color. And then secondary colors, they too will be detected by these stimulus values. For example, if the orange monochromatic light with a certain wavelength, it strikes the retina, it will stimulate red cones with a stimulus value of 99%, green cones with a stimulus value of 42%, and blue cones not at all, 0%. So this set of stimulation of cones, 99%, 42% to 0%, this set of values will be perceived as orange monochromatic light. Or let's say yellow, it has a certain wavelength certain wavelength of light, and yellow color will simulate red, green and blue cones with a stimulus value of 83%, 83% and 0%. So this set of stimulus value 83, 83, 0 stimulation of these respective cones is perceived as the yellow color. So that's the basis for Young and Helmholtz theory for color vision. Now that being said, once the cones have been stimulated with a certain stimulus strength, then the further processing will take place in the visual pathway. So you can see the diagram here, ganglion cells in the retina, they will convey the fibers to the lateral geniculate nucleus. You know, in the lateral geniculate nucleus, there are two laminae, magnocellular and parvocellular, and between those two laminae, there is interlaminar region. So this interlaminar region is where mainly the cones will convey their stimulus values, and then in the visual cortex, this signal from the lateral geniculate body to the visual cortex, the signals will be conveyed. In the visual cortex, there is an area called as blobs, and this is responsible for the color vision. So eventually when the signals reach the visual cortex in the blobs, color will be finally perceived. So you have to discuss about the cones and different stimulations of different types of cones, primary colors and secondary colors, how they are processed by the cones. Then you talk about the lateral geniculate nucleus, and finally the visual cortex. That is how the color is perceived or colors are perceived. The second part of this video is on the color blindness. Now, defect in the color vision or complete absence of a particular type of color in an individual. So abnormality of color vision is inherited as X-linked recessive disorder. So it's a genetic abnormality, and or it can also be some kind of an acquired defect. Let's see what those defects are. But depending on whether a person can perceive three colors or two colors or only one color, the nomenclature is like this. A trichromat, three colors. He or she can perceive all three colors. So a normal color vision. Then, so far as the defects in the color vision are concerned, there are dichromats. Dichromat means two colors can be perceived. That means the third one is absent. So one cone absent or defect in one type of color. That's dichromat. Now, as you know, you might be aware of this, that the colors have been given numbers. So red color is given number 1, green color is given number 2, and blue color is given number 3. And based on that, these defects are called as protanopia. Protos means 1. That means color number 1 is absent. What was it? Red color. So therefore, red blindness is called as protanopia. Deuteranopia. Color number 2 is absent. Color number 2 is green. So green blindness. And the third one is tritanopia. Color number 3 is absent, that is blue color absent, so blue blindness. Okay, also you must understand that opia will be a complete absence of that color vision, whereas anomaly means weakness of that color but not complete absence. For example, if I say protanomaly, it's a weakness to perceive red color. It's not a complete absence, but red color is not perceived entirely normally. Weakness for the red color would be called as protanomaly, and similarly deuteranomaly and tritanomaly. If you want to increase the length of your answer, you can again write those anomalies as well. And then we have monochromats. Monochromats means they have only one color present. That means two colors will be absent, two other colors will be absent, and two cones will be absent. Most common in this is the red-green color blindness. That's the most common type of monochromatism. Now coming to the acquired defects, and this is Physio plus plus. Normally this is not there in the answers of most of the students. So if you write this, you will get extra marks. That is acquired defects in the color vision. Drugs and their side effect being defective color vision. Digitalis. You know, digitalis is a drug that is given in heart failure, but its one side effect is that it causes xanthopsia. Xanthos is yellow, so there's a defective yellow color vision, or there's a problem with yellow green distinction, confusion between red and green to distinguish between these two colors. These are temporary side effects. As the drug is discontinued, these side effects they just go away after a certain period of time. Sildenafil, common trade name Viagra. This is known to cause blue vision. I mean, visual spectrum shifts towards the blue wavelength, and there is blue tinge to the to all the colors, which is called as synopsia. Cynos is blue, so synopsia. And the third drug is ethambutol. Ethambutol is an anti-TB drug, and it is known to cause weakness or defective color vision, and therefore when these drugs are being given to a particular patient, they are advised not to operate the machinery or not to drive the motor vehicle because they have to see the traffic lights. Right. That was about the color vision defects. And finally, the tests to detect the color vision. As I've already mentioned, when are these tests required? They are required in certain industries which are dealing with various colors, or while operating a motor vehicle, you need to have a clear-cut color vision so as to see the traffic lights. Okay, now there are simple tests, but the most commonly used and which you will have in the practicals is the Ishihara test. this Ishihara test consists of pseudo isochromatic plates. Pseudo isochromatic plates. There are 38 plates in all in that booklet which is called as Ishihara's chart. Now in this, why are there so many plates? I mean, you could have just one or two plates, and if a person is able to identify the three colors, there are numbers, dotted numbers. You might have seen these plates. But if you have to see whether the person is having normal color vision or abnormal, means there is defect in the color vision, then you will have to have at least 6 to 7 first color plates where the numbers can be read by the person. And then if it's a color defect, at some plates there are no numbers, just some arbitrary figures or arbitrary sketch by those drops. But a person with certain color blindness identifies some number where a normal person cannot see any number. So based on such principles is the Ishihara's chart. I'll make another video specifically that deals with the Ishihara chart and how we can detect the color defects or color blindness. But this was the color vision and color blindness. You might get a long question where you can write all this. I'm putting this these notes in the comment section. So that's all for a long question on color vision and color blindness.